mute, so
yeah.
There you go. If you want to turn it on and off, there you go.
Yeah.
Okay. Well, I'll give a few seconds to get folks sort of situated, and then we'll go ahead and get started.
Hey, George, it's Marc Andreessen. Thank you for joining us tonight.
Oh, thank you, Mark. Great to be here.
In another world, we would have been doing this probably in a real stage with you and Mark to talk about all things, where biology and technology are intersecting.
Yeah, which is almost everything in my world.
Exactly. Excellent. So, look, I think a couple minutes in, why don't I go ahead and get started? So thank you all for joining us tonight. Welcome to It's Time to Heal. For those that have joined us in the past, you'll know that Time to Heal is our Clubhouse room at a16z to cover the future of bio and healthcare very broadly. And what we aim to do here is to have a very loosely structured and interactive discussion. So for folks that I may not know or may not know me, by way of brief introduction, I'm Jorge Conde. I'm a general partner here at a16z. And with me here tonight, I'm fortunate to have my other GP colleagues, Vineeta Agarwala, Marc Andreessen, Vijay, Pande, and Julie Yu. Today, our special guest is my friend George Church, who is a professor of genetics at Harvard Medical School. George is also a very prolific founder of startup companies that emerged from the work in his lab, many of which are represented by some of the folks in the room here tonight. And I myself I had the opportunity to co-found a company with George almost 15 years ago, which I think aged George horribly. So George, thank you for putting up with me so many years ago. And certainly thank you for being here tonight with us. I just want to put a quick note out there for all the folks here that this conversation is being recorded. For those of you that are interested in come up and chat or ask questions later on in the show, please note that by doing so, you're consenting to us using your words and profile image in a future recording related to this event. Okay, so why don't we get started? George, we've known each other for a long time, and I think one of the things to me that's always been fascinating is how you think about the future and really what the future will look like and what, the impact of advances in science and technology will help, how they will help shape that future. And about almost 10 years ago now, back in 2002, you wrote a book called Regenesis that described how synthetic biology would reinvent nature and would reinvent us as a species, as humans. And it makes me think when I read that book, it made me thought of something that people attribute to Bill Gates, what they call Gates' Law, which was a quote where he said, "Most people overestimate what they can achieve in a year and underestimate what they can achieve in 10 years." So looking back nearly 10 years later after writing this book, what do you think you got right and what do you think you got wrong about how synthetic biology would shape us and the world around us?
Well, fortunately, I didn't make too many predictions about timing. Next year or 10 years from now or 50 years, but everything is going faster. Let's start with what I got wrong or didn't anticipate. Actually, just 2 years later, I did a paperback version and had a chapter at the end on eight things that weren't covered, including little things like CRISPR that weren't covered in 2012. But what I think that my biggest surprise since then was protein design. So I, as a teenager, did energy minimization with coordinates, and David, my good friend and colleague David Baker, has really run with this, and we've run with him on it. And so I was a little surprised when some of my people said, well, let's do machine learning on this without using coordinates or energy functions. And I said, oh, that sounds interesting. I've got 40 years to unthink. And it worked like a charm. We just published 4 papers, one with Google, and started 3 companies that were based on this which is Nabla, Manifold Bio, and Dino. And it works like a charm, just using sequences, plus the other key ingredient, in addition to lots of sequence data, lots of machine learning, is lots of synthesis. So so anyway, that was something I think you could say it's wrong by omission. It was not mentioned in the book, Regenesis. Something that was intermediate wrong was chirality. The book, it's one of its themes is being able to make a mirror image world. We haven't done that in 10 years. I didn't say we would. We have made progress, and we'll hopefully get our first couple of papers are starting to come out already. So that's good. And
by the way, it's probably worth to just take a second to describe why is chirality? Why is the mirror world important?
Right. I guess it's obvious why protein design is cool and machine learning. But yeah, chirality is a way to make. By changing the genetic code, we think any organism resists all viruses, and that is actually also mentioned, and that is working out great. But with chirality, changing the handedness of the molecules of the cell, you can make them not only resist to every virus, but to essentially every pathogen, every enzyme, and you can make a whole new set of chemistry, which is all the chiral and all the mixed chiral centers and so on. So that's why chirality is important, and we thought we had a shortcut, and every day it gets easier and easier. We see new paths, not shortcuts so much as pathways through it. So anyway, I think that's somewhere intermediate between not completely getting it right. You might imagine we were more optimistic.
But George, arguably those are details, right? Like, what about the more big picture stuff? Sounds like maybe that's more right than not.
We'll see. I mean, it'll be a while before we see whether it's useful and so on. But one thing I think we did get right was there was a big emphasis on multiplex editing. In fact, in a certain sense, multiplex everything, and that's turned out to be true. We now have a lot of multiplex editing going on. At least a few edits are used in CAR T therapy, but more importantly, we've now got pigs that have been engineered in 42 different places, called Pig 3.0, and there's 2,000 of them now that are alive and breeding and having a good time, except when we take their organs and put them into preclinical trials.
So you kind of got CRISPR, right? You didn't.
We've done many different kinds of editing before and after CRISPR, and certainly have started some nice companies with CRISPR, including eGenesis, Editas, Intellia, and Inari for agriculture. But Tessera is an example of a non-CRISPR transposinase-based new startup flagship that I've been involved in. So I think, the point is not just editing one thing, which is what most people are doing with the editing, but editing multiple sites in the genome. I think that's starting to take off. Our record so far is 22,000 edits in a single cell. Pretty much on target.
So if you were going to sit down and write the book, well, first of all, I should mention a factoid for this audience. This book had the most physical copies in the history of books, correct? Because you encoded it in DNA.
Yeah, still does. Yeah, 70 billion copies. Yeah.
So how did you get 70 billion copies of this book published?
Yeah, well, we encoded it into DNA, and then it's very inexpensive to make copies of DNA. Actually, Sri Kosari, who's on I can see his icon on the screen here, was my mentor for that. He was the PI and I was the postdoc, and that was maybe something I underestimated where that was going to go. It still hasn't delivered something of great use to society, but it's certainly picked up a lot of momentum with the likes of Technicolor and Microsoft, and now a whole consortium involving Twist and so forth, that seems to think that it's the next big thing, but we'll see.
So if you were going to sit down today and write the book again, right? So if you were writing the 10th anniversary edition, you mentioned you updated it for the second year after the paperback came out.
Yeah.
What's the mind-blowing thing that you couldn't have imagined when you wrote the first edition that you would write into the 10th anniversary edition?
I wouldn't say that machine learning for protein design is quite mind-boggling, but it's probably the biggest surprise that I had personally. I think where multiplex editing is going with making any organism resistant to all viruses, I'm more confident about that than I was. But again, if you've read the first edition, you'd be disappointed if that's the mind-boggling thing 10 years later, even if it is. So I have to say I don't know what it is. I am writing a book, which actually I started before Regenesis, but it's so wacky and I can't really say much about it. But anyway, there will be a book.
But I know you
say
can't say much. Go ahead, Vineeta.
No, I was just going to say, I can't believe we skipped over Pig 3.0 so nonchalantly. You got to tell us what 2.0 is and how those pigs are doing. And a little bit of context might be really interesting for the group on the goals of that project.
Yeah. Well, thank you for helping me communicate. So the 2.0. Well, so this is an old field. The idea of xenotransplantation goes back at least 20 years, and some of the originators, pioneers of the field, hoped that changing one gene would be enough. There was one gene that had a huge impact, which is alpha-1,3 galactosyl-transferase. But that was not nearly enough. I mean, that got us from rejection in a few hours to rejection in a few days. But over the 20 years, everybody worked on their favorite gene, and they accumulated a long list of genes, their wish list. And the pioneers invited us when we co-invented CRISPR to join in and edit multiple genes. And so it got to 42 genetic changes by 3.0. 2.0 was a boring intermediate. It was sort of halfway between 0 and 42. But at 42, it's basically everybody's genetic wish list all in one pig. And like I said, they're healthy. There's 2,000 of them. You don't get to 2,000 if they're not healthy, which is kind of and this is all germline, by the way, and it will be in humans. So in a way, humans will be benefiting from germline CRISPR, whether that's what we meant when we say that it's controversial. It's not what we meant. But anyway, they are in clinical trials, preclinical non-human primates at Massachusetts General Hospital. Jim Markman is heading that team, and it looks good. 300+ day survival with sort of normal immune suppression. One of the problems with the early xeno was that they would use immunosuppression that wasn't approved for humans.
So, a question just to pull on this thread a little bit. You've been working on versions, pigs 1.2.0, 2.0, 3.0. Xenotransplantation as a field has been around for decades, as you said. It's obviously made great advances recently with some of these new technologies and approaches. What are something, you've been in the synthetic biology field, you've helped create this field. What are some of the things that work in synthetic biology that, not to sound simplistic, but that you can't believe they actually work? And what are some things in this field that don't work yet that you can't believe we haven't figured out how to make work yet? Because I think for a lot of folks externally, they don't realize how hard biology is. Things take time. So I'd love to just get your sense as someone who's on the bleeding edge of what what's surprising that works and what's surprising that doesn't work yet?
Well, so one of the things that doesn't quite work yet that is a really good homologous recombination, where we get precise editing, where we can swap in big chunks of DNA into, say, human cells. Even though the very first paper we published on CRISPR did involve homologous recombination, it was at sort of 8%. And you can do better now, but not that much better. And there's a lot of incorrect things that happen along with that 8%, a lot of genome vandalism, I call it. So that's one thing. The other thing is even more similarly disappointing so far. All these things are so far caveats. Is what one of the things that was considered part of the dawn of synthetic biology, not really my version of it, but still something that's recognizable, is genetic circuits. There's this whole metaphor with comparing synthetic biology with electronics and computer science, and we all wanted to make circuits, and it still is hard to make circuits that do something, these kind of sophisticated circuits with multiple levels of even basic Boolean logic haven't produced much useful yet, but I'm sure that may have already changed and I didn't notice. So those are two things that I'm not sure. It's not that I wouldn't believe that they don't work yet. I can believe it. It's just disappointing and a little bit surprising.
George, do you think it's an issue of it not working, or is that it's an issue of that's not really useful for doing something right now, even if it could work?
Yeah, well, there's a fine line there in my. If it worked really well, there'd be useful things you could do with it. So the fact you can't do something useful is indicative that it's not working well, and vice versa. So, yeah. I think the thing that is mildly surprising, again, it's hard to say that you're totally shocked about this, but is how well libraries work, synthetic libraries. I mean, I've come to think of synthetic libraries as kind of like a computer peripheral. It's like quantum computing. It's if you want to do a really hard problem that like figure out how to target adeno-associated viral vectors to one tissue and not all the other tissues, then you might use these synthetic libraries. So rather than random mutagenesis, you do highly directed. And we just published a paper a few days ago where we showed that we can make up to 29 missense mutations simultaneously without killing the virus. While when you do sort of random mutagenesis, it's hard to do more than 4 at a time. So I'm sure some people in the room will be shocked by that, and some won't. Yeah.
So what you're saying is that, the tools are getting better, so therefore, this is probably an obvious conclusion, the capabilities are therefore getting better, so the things that haven't worked yet, but may in time as our capabilities improve.
Yeah. And the other thing that's a little bit surprising, you asked me, sustain this exponential. It's going quite a bit faster than Moore's law by a few measures, and so you might expect it to burn out faster, but it doesn't really show much signs of letting up. Ultimately both of them fail when you start getting to an atomic scale, but since there's such high parallelism, it's so inexpensive to make trillions of members of a library. It's not so easy to make trillions of chips or even components on chips.
So talking about advances, one of the comments one of the arguments I've heard you make in the past, which I found counterintuitive, but also very compelling, is this idea that in this field, in your field, we see advances, a lot of the advances that we see happen like in these constrained moments in time, where you talk about how when we had restrictive environments like moratoriums for recombinant DNA or embryonic stem cells, the field was still able to advance in spite of, or maybe even because of, that restricted environment. And so we've been in this pandemic now for, call it a year. And on the one hand, well, not on the one hand, I think it's been remarkable that we've seen the speed by which we've been able to develop vaccines, against COVID-19 or SARS-CoV-2. We have 3 authorized vaccines now here in the United States. 2 of them are on an entirely new platform, right? And the mRNA, there are mRNA-based vaccines. Do you think, number one, is it pretty obvious that we wouldn't have these vaccines this fast if we weren't forced to get there because of the pandemic and the situation that it's caused? And number 2, do you think that the protracted lockdowns and, all of the other, sort of factors around the pandemic might have other effects that aren't obvious yet in terms of advancing the field or advancing science?
Yeah, it's definitely a time of revolution. What happens when an asteroid hits Earth is you get an evolutionary revolution. The same thing is going to happen with something. You just, by every definition, this is a revolutionary time. It's a very unfortunate and very expensive experiment that we're doing. It's probably $16 trillion, maybe just for the United States alone. The United States is probably going to be the hardest hit, would be my guess. It looks like it's so far about 400 times more deaths per capita in the United States than in places like China. And so we're learning lessons. It could. I think we're going to, inevitably, we biotechers are going to pat ourselves on the backs for vaccines and drugs and other high-tech things, high biotech things, diagnostics that use all kinds of creative enzymes. But the real revolution is in masks and social distancing. That's what saved all the countries that did the best. You also need vaccines, definitely. But if we were to take something home, in fact, the masks we had have been terrible for the most part, but using them would have been better than nothing, and we should have had better masks. I mean, this is not rocket science to make a mask that actually seals around your neck and has HEPA filters. Better than the N95s, but anyway.
So for the folks that don't follow you on Twitter or on Instagram, and by the way, if you don't follow George Church on Twitter and Instagram, you should remedy that now. But you posted a photo just the other day on a mask, and it's basically a mask that you can see your entire face, and you had some sort of straw in your mouth. What's the backstory there?
Yeah, that's microclimate, it does what I had been trying to do, which is makes a seal around the neck where people have almost no hair, and a good seal, and it has full facial availability, which is a thing that you want, and it has 2 input filters and 2 output filters, and those, in principle, those filters could be much higher quality than the ones that are in N95 masks. But the main thing is having a good seal around it, and the batteries last for 8 hours, and it's great for flights.
Did you make it? Did you make that mask?
No. I made prototypes that were essentially competing with it. But it's way better than anything that I did. But it's the same idea, sealing around the neck and having the whole face visible. And
you can eat and drink with it on, I saw.
I think that's an important thing to do. That isn't one of the things that they stress, but I think it's important to be able to do that, because if you take your whole mask off to eat and drink, you kind of defeat the purpose. But yeah, you can definitely, as long as it's in liquid form or you have some other way of sneaking in solids. That's not the perfect thing. Also, there's a little, it's a little noisy, so it's better for reading and working and sleeping. But you can have good conversations. It's perfectly possible once you get used to it.
And so, at the beginning of the pandemic, sorry, go ahead, George.
No, go ahead. At the beginning of the pandemic.
I was going to say, at the beginning of the pandemic, it was reported, and there was this great article with a great photo of you giving yourself a DIY vaccine. So number one, were those reports accurate? Did you in fact, vaccinate yourself with a do-it-yourself COVID vaccine? And if so, have you, or would you still get sort of one of the official ones now? Like, do you consider that you were vaccinated with by that, like effectively, or are you still going to get one of the official ones?
So that was a very small study called RADVAC, the brainchild of Preston, Estep, and others. And the preliminary data is that there seems to be some immunity in the mucosal nasal sector, but not in the blood. So it would not be recognized by the standard blood test. On the other hand, you might imagine that nasal and mucosal immunity might be a good thing. So yes, I was vaccinated. We went through 9 generations. It's very, very fast generations. Many of them were within a couple of weeks of a paper that would come out. So paper comes out on T-cell immunity updates, and we could update the vaccine. So these generations were happening every few weeks. So that was a positive side. Very inexpensive, about 25 cents a dose. But of course, that would go up with the cost of clinical trials and marketing and so on. There was surprisingly little effort on. I mean, I don't think this is a done deal yet. I am taking the conventional, now quote conventional, messenger RNA vaccines very soon, and I'd be happy to take them all eventually once everybody else gets theirs. Very much pro-vaccine. I've had the rabies vaccine, for example, because I had a close encounter with a bat. Love vaccines.
And what was in the version of the DIY vaccine you took?
So the 3 innovative things were the nasal delivery. So only about 5 out of 300 of the initial vaccine trials were nasal. Second, it was the carrier adjuvant was chitosan, which is a common polysaccharide in many arthropods like shrimp. And the third thing, it was you could customize the peptides involved, so you can avoid peptides that might cause antibody enhancement. You can focus on the ones that maybe would be buried or not properly displayed, etc. I mean, in principle, it has advantage. Doing something that is as close as possible to the natural virus isn't necessarily the right thing if the virus has been gaming the system. So synthetic can be better. In any case, it was just our shot on goal, and time will tell. I hope that COVID is as easy as smallpox, polio, mumps, measles, and so on. But it could be as hard as HIV, tuberculosis, and malaria. We don't know.
So actually, on that topic, do you think, do we have a potential, maybe silver bullet is obviously overstating it, but do we have a potentially important new weapon with these mRNA vaccine technology to go after these viruses that have evaded our efforts to make vaccines, or is it, it's the complexity of biology, and yes, it's a new technology, but it's not going to be the obvious thing that helps us solve those unsolved problems?
I think it's totally justified that we're giving messenger RNA therapies and vaccines their day in the sun. Sunlight is the best way to figure out whether something is good or not. But I don't think it's established yet. I don't think that suddenly, if we started using messenger RNA on tuberculosis, malaria, and HIV, they're going to suddenly cave in necessarily. I hope so. That would be great. But it's just another way of doing the delivery of the antigens.
Got it. So if I could, I was going to shift a little bit. We've been in this pandemic for a year. One of the things that you're most well known for is your incredible lab at Harvard Medical School. First of all, is the lab functioning right now? Or you guys, do folks go into the lab, or is everyone working remotely? How are you managing through this?
Well, we were fully operational throughout. We were never down because a fraction of the lab was working on COVID-19 diagnostics, therapeutics, and vaccines. Number one, and number two, a huge fraction of the lab has always been computational, so that can be done pretty much anywhere. Since early summer, we've been doing shifts, because we had a big lab when they did return, when the people that weren't working on COVID returned, we had to do 2 shifts. Now we're trying to transition to 1, but our lab was already at pretty high density.
So speaking of high density, one of the things that you're very well known for, as I said at the outset, was how prolific your lab is in spinning out startups. I think it was back in 2019, or it might have been 2020, Nature Biotech published a news feature on you and your lab. And the news feature was titled 1 year, 1 lab, 16 spinouts. And it had, in sort of the lead there, it started out by saying, in 1 year, George Church's group spun out 16 different startups. What explains the lab's incredible output of entrepreneurs? ? Now, the problem is Nature Biotech is behind a paywall, so I don't know the answer. So can you give us the answer? What explains your lab's incredible output of entrepreneurs?
I'll send you a copy of the answer. I don't remember their answer, but I would say my answer would be, the first, immodest thing would be to say it's me, and I can say it's not me, or at least not me in 1986 when I started the lab. I think I was particularly ineffective, and most of my ideas were about 100 degrees off, maybe 179 degrees off. And most of the people in my lab thought I was completely nuts, talking about nanopores and nanogrids and sequencing all the people in all the organisms. And they were probably right. But anyway, I think what might partially explain it is the concentration of good people that comes just from being at Harvard and MIT. So, I'm lucky enough to get PhD candidates from both schools and multiple programs. So, and then that acts as an attractant for good postdocs as well. And there's a kind of a positive feedback loop that starts. And when you start, compounding interest since 1986, eventually you get kind of a beacon that and maybe a few lucky breaks, next-gen sequencing and next-gen synthesis and next-gen editing, and a bunch of multiplexing and libraries. And anyway, it's a positive feedback loop. I think that's the simplest explanation. Also, I think, that's probably it.
But it's interesting because, if I and I know we as a group here have had the good fortune of getting to know many of the folks that have come out of your lab, and in many cases, I've had the opportunity to work with them and even invest in them and support them. One of the things that I find incredibly consistent when you talk to Church Lab alums is, how they credit, sort of the audacity or the ambition of their vision or their ideas to their time in the lab. And so, and the question for you is that something you're selecting for when you're looking for people to join your lab, that you have the benefit of these incredible institutions that are around you to select from and to pull talent from? Or is that something you're teaching in the lab, or is it both?
Well, it's both, obviously. I mean, that's a setup, but it is, it's not something that necessarily I teach, but the lab teaches. It's kind of each generation, it gets a little more audacious, and they teach each other. And the alum come back and teach us. They're like older siblings that like their younger siblings and teach them stuff. And it's just wonderful to watch this encouragement that occurs. And to some extent, I think the culture that we have is not one of think, hiring a bunch of geniuses and thinking about something until you have a perfect experiment and publishing it. More a matter of taking a fantastic number of shots on goal. So it's not like if I had to choose between the one perfect experiment and doing a million failures with maybe one success, I'd pick the second one, because that means you've actually winnowed through a lot more interesting, almost interesting things. So it's more trial and error, and that's kind of embodied in the whole libraries. That if you make a library, instead of testing one hypothesis, you can test a million or a billion at once, and that's essentially what we do both in our multiplex experiments and in our life.
Is one of those multiplex experiments that you do? You're gonna accuse me of giving you another setup, but I promise.
Okay. If I just stopped with the word both, that would have been cheap for both of us.
Fair enough. So here's another setup. You talk about multiplexing experiments. How do you decide how do you all collectively decide what is the work and provenance of the lab versus what should be spun out into its own company? Like, is that something that's deterministic, or is that something that just sort of emerges organically?
Well, there's a little organic. There's a little bit case by case. I think we develop a kind of a collective intuition as to when something is too early, and we try not to be ahead of our time too much, anyway, because that's not as cool as it sounds. But with a lot of our stuff is aimed at bridging pure science that we do with technology that we, kind of crazy technology with some societal benefit, medical or commercial, what have you. And so they're almost always destined to be to go that way. And the postdocs that are co-patenting, the key to having a big flow of companies is to have a big flow of patents. And the postdocs that patent it, when they patent it, they already know that it's moving in that direction. They just have to assess whether they would prefer to work on their own invention or go work for somebody else's company. And increasingly they've been voting for themselves to run their own companies.
And we're very lucky that they have been, because I mean, just the flow of talent has been incredible.
Yeah.
I mean, George, I'd love to jump in and ask you, what would you like to see other universities do or other labs do to sort of try to improve that flow of talent? I'm curious to get your take there. I mean, everyone's going to do things different ways, but are there any systemic changes? that you'd like to see?
Well, this is actually a fairly frequently asked question. We get a flow of people. Not to say it's not a good question. We get a flow of people, big shots coming in to figure out what's the secret sauce at the Wyss Institute or in my lab, and some of it I think you can take away with you in your briefcase in your head, but some of it I mentioned. Alluded to is the concentration that occurs. There's something to be said for anti-elitism, where we spread the wealth evenly everywhere. There's something to be said for really highly concentrating, making a room full of the best and brightest, or making a university full. And so having Harvard and MIT in such close proximity, and they really are amazingly close. They're actually intertwined in that there's a bunch of us that have appointments at both universities. While I've been, I was a postdoc at UCSF, and I participated in that culture from Berkeley to Stanford, and it's a lot further from Berkeley to UCSF to Stanford than it is between Harvard and MIT. They have programs like QB3, which I think was a good idea, is a good idea. Mm-hmm. But anyway, so that's the hard part about taking it away. But some of the other lessons, creating an environment that tolerates and even encourages failures, millions of failures, where you just bounce right back, or you do them all in parallel and something comes out.
Those are the multiplex experiments now on a lab scale.
Yeah, I think that could be taken home by many groups, no matter where they are.
Do a quick shameless plug, but George was nice enough to sit down with me on our a16z podcast. I think it was almost exactly 2 years ago to this day, where we talked a little bit about that that magic. The name of the episode was "What's in the Water in the Church Lab," for that very reason, because it's like one of those things. It's hard to, it's really hard to, articulate and certainly very hard to replicate. And it's been a remarkable thing to participate in and to observe from a distance.
Yeah, we're certainly not trying to keep it to ourselves. If to the extent that we don't understand it, we're still welcoming people to come visit, and if they can figure it out, that's great.
What are some of the ideas? Again, to just stick on this theme of experiments for a second. And you talk about generations of students coming through, and you gave the example of Sri Kasuri sort of teaching you about how to publish your book in DNA. What are some of the areas where you feel that you're pulling the lab, and where are some of the areas where you feel like the lab is pushing you?
Well, I think the best example of the lab pushing me is probably the machine learning.
George, your audio just went a bit wonky. All right. So I think while we're waiting for him to come back, maybe this is a good time to start to invite some folks that we can join us in the conversation. And maybe if I could, one easy place to start is with Sri Kasuri, who I do see in the audience. So Shri, we're going to ask you to come on up. There you go. And while we're waiting for George to come back online, I guess the question for you, Shri, is how accurate was his telling of the story in terms of you getting Regenesis published in DNA?
Oh, that story, yeah. There's George. He's back.
What's that?
Hey, George.
You went silent for a few minutes.
Oh,
yeah,
that's weird.
Your audio was wonky.
Okay, is this okay now? No?
Yeah, it's okay now. But now Shri's up here with us, so he can respond to us. So now we got to be gentle.
Yeah. Well, anyway, that's all I had to say. Yeah, I'm going to pull a Forrest Gump here. That's all I had to say about that. You had to be there.
That is the first time I heard George's Forrest Gump impression, quite honestly, which was amazing. So let's see, Regenesis. Yeah, George, I think that the background on that story, it's a little bit funny. I think I was going around Harvard, MIT, trying to find like good things to build in DNA, and no one had like enough designs to like kind of satisfy the scale of buying one chip, one oligo array that cost a lot at the time. And so I went to George and I'm like, can I just build Moby Dick in DNA and just get it over with so I can just prove the like synthetic methodology? And George, which is the weirdest thing to come out of George's mouth, was like, no, you should build something more practical and useful. And it slowed things down. Anyways, like long story short is like, you know, we ended up building normal stuff, but at some point this project laid around this like, and George and I had talked about it. We talked about some encodings. And at some point, George wanted to get back in the lab for one reason or another. And I'm like, oh, you should just do this project. Yeah, I think you cleared out 2 weeks, George, of your schedule on that.
Yeah.
And came to my bench. It was super cool to see because George, I don't think you had ever seen like or operated real-time PCR up till then. And like you were like sitting in front of the machine watching your PCR go, which was fun to see. Yeah, he did.
Well, the idea of you introduced the idea of stopping it before it went too far, which I thought was beautiful at the time.
Yeah, that was I think ideas that had permeated the lab before I got there. But I think the bigger thing is like we finished the thing. You know, George wrote up the paper. I edited it a bunch, and we submitted it, and it just went really easily. Quite honestly, like that paper went super well, and I think it created a false sense of like power in George. It's like I can work for 2 weeks and get a science paper, and like what are all you morons doing?
I didn't say moron.
That's how we all felt internally at the company. But yeah, no, it was a fun time. It was cool to be on the background of that.
Yeah, a little bit more backstory, just very briefly. Part of that project, not the part that Shri was talking about, but the other part started when I wrote a review of a paper from Craig's lab, Venture's lab, where they had put a code into mycoplasma, and I wrote the review in DNA and sent it in, and the science editor couldn't read it, of course, and passed it along to the authors without filtering. And I had written it in a different code than the one that they had used, and then I thought my code was better, so they had to decode it. And I think it was Clyde Hutchinson who was good enough at computers to figure out how to decode it. But anyway, that's the kind of code that we use for the book as well.
So
yeah,
George,
I forgot about that.
As you can see, we're inviting up a bunch of other folks, so this may inadvertently turn into a George Church roast, and I can't be held responsible for that.
That's okay.
I will say, Jorge, one comment on the atmosphere in the lab. It definitely feels very different, but I do think some of it was, it definitely felt like a motley crew, at least when I was in the lab. It wasn't like people with great resumes necessarily. Like I, for one, was fired from one of George's companies that he co-founded, and I was the first employee of, like, in a bad firing. Like I was like walked out off the premises, and I literally started in George's lab like I think 3 or 4 days later. So like I think it's I don't know, it's definitely filled with, I think George's words at the time were about neuroatypicals and things like that. So a lot of intellectual diversity in the lab that was powerful, I think.
By the way,
should we get a family reunion going here?
Let's do it.
If you're a Church Lab alum, raise your hand in Clubhouse, and we should be able to get you up here.
While we're waiting for people to come up, I'll just follow up on Shri's last story there. When I first met him, and we were talking about his new company, which we are proud investors of, and I'm proud to serve as a board member of. In our first conversation, Shri said, hey, just so you have some background on me, I was walked out of my first company by in a very bad firing. I'm going to tell you that full story because I want to make sure that we start, with very open and clear and transparent communication. It was remarkable, Shri. So thank you for doing that, for sharing that. Okay, who else do we have here? Eric.
Yeah, here. Hello.
So Eric, I believe George mentioned you in the context of some of the work that they were doing around protein engineering.
Yeah, that's right. We started a company that George also mentioned called Dino Therapeutics, and we're focused on engineering AAV capsids for improved gene delivery. And that's basically combining the multiplexing that George spoke so much about, plus machine learning and a kind of exciting next-gen directed evolution workflow.
And what years were you there?
So I joined George's lab. Actually, also another funny story. In, I guess, February of 2015. And this is a good example of George giving people an interesting opportunity. Basically, I pitched George on what must have been a really exciting idea to come in the lab and hang out for about 6 months and then to leave the lab and go somewhere else. Because I decided at that point in time I wanted to go into industry to join a startup. And I didn't really have many skills that I felt would be marketable to startups at that time because my PhD was really applied to basic biology. And I didn't know anything about systems that would be relevant for industry. So George was on my advisory committee, and I had some stuff I needed to finish up from the PhD. So I pitched this great postdoc idea of coming in for a few months and hanging out and meeting new people and maybe transferring some knowledge to the lab. And in the end, found a really cool idea to apply these multiplexed next-gen synthesis approaches to AV capsids, and then came up with the idea of doing that supercharged by machine learning. And then that seemed like a pretty good idea, so I ended up staying in the lab for 4 years. And then 4 long years later, we started a company around it, and we've done really amazing things in the last 2 years since then. So it's one more example, I think, of George giving people a chance to try something atypical, and then in this case, it worked out really well.
Hey, Paris and Alex, it'd be great to hear your voices too, as relatively recent alumni of the lab. George said each generation gets more and more ambitious. So curious how you had a chance in particular to interact with alumni and how they influenced you.
Yeah, so this is Paris. I'm currently the CEO and one of the co-founders at Dino Therapeutics. And of course, from the name, you can guess that how much we feel we are still attached to GC and like the church lab. We definitely we started from George's lab, thinking about, or maybe I should also start from my background story when I met George for the first time, which was during a Wyss retreat when I was doing a postdoc. And I always say that George sees what you really don't see in yourself. In my case, for sure, that was one of the most important things that I had during my first interaction with George, because I started talking to him and I told him about my background because it's in tissue engineering and neural engineering. He told me that, oh, that's a very interesting background. Do you want to reach out and we can talk? And at the time, I really didn't think that I have the right skill set to join George's lab. But interesting enough, this is one of the unique things about George and how open-minded he is about his lab and the lab members, because I had a lot of entrepreneurial background. So I definitely immediately felt that I fit right in, and right away started connecting to the network of alumni, including the folks at Regenesis and other groups from the lab. And really early on learned that, okay, this is what we want to do. I want to continue the entrepreneurial passion that I have. And I asked George what project he recommends that I join based on my background. And he strongly recommended that I join Alex's TF4 project that then at the time, it was in 2015, that we both started seriously thinking about commercializing it. And then now here we are 5 years in or 6 years in, and we are on our way to commercialize the technology.
So that's fantastic. That the array, I mean, the broad array of just of projects, of technologies, of companies that have come out of the lab. I mean, it's incredibly diverse. And when